Alrighty, hi guys. Welcome to my video, Whole Organism Cloning and Therapeutic Cloning. So this is the second half of week 13. The first half was a CRISPR-Cas9 gene editing, and that one I covered in a previous video. So this is going to be the second half of week 13, where we address methods of whole organism cloning and therapeutic cloning. So hopefully this video will be helpful as we walk through the various techniques of how they go about this and their usefulness. We're not going to talk so much about the ethics because my job is to show you how we do it, and then you get to decide for yourself whether we should. So that's the fun part about this week and next week is we get into kind of ethics and the debate about that. So that's always a fun conversation to have on the discussion boards. All righty. So there are two main types of whole organism cloning, but first off, let's just clarify what we're talking about here. We use this word cloning in genetics quite liberally, and just because we're scientists, we have to make things very, very difficult and confusing. Back in week 10 and week 12, we used this term cloning as part of the term gene cloning. Now, gene cloning, as we learned in weeks 10 and 12, is the process of taking a gene from one species and putting it into another species to give that second species a particular trait that it didn't have before. Putting it into different species. Now, in contrast, this week we're talking about whole organism cloning. Man, I can spell today, I promise. Organism cloning. And this one's different. Rather than taking a gene out of one species and putting it into another to change that species, to give it a new trait, in this case, I'm trying to take an individual and make an entire individual that's 100% identical to that person on a genetic level. So this one is like Star Wars Attack of the Clones. I'm trying to take a person and make identical photocopies of that person. So rather than just moving a gene over to change a species and give it new abilities, I'm trying to completely copy an entire person and make them 100% identical to the person they were copied from. That's different. Let's see here. Make 100% identical copy of an individual. So this week we're talking about whole organism cloning, and that's what we're going to cover here in this video is how we can go about doing whole organism cloning and what its uses could be. So under the category of whole organism cloning, there are two main types that we use: artificial embryo twinning. And the second type is called somatic cell nuclear transfer. kind of a mouthful there. So we're going to talk about each of these two different types and what the differences are between them, and when you would use each type, because there are different applications for each one. So let's talk about how these two work here. First one we're going to talk about is artificial embryo twinning. Artificial embryo twinning. And the idea behind this one is an embryo is a developing it's a developing child. So after the sperm fertilizes the egg, it forms what's called a zygote. That's a fertilized egg. You don't need to know that for this class, but then that egg starts to divide and divide, and it forms this little bundle of cells, and then it grows and grows until it becomes a baby. But while it's growing as that bundle of cells is gradually turning into a baby, it's called an embryo. Now there's in nature, we have something called identical twins and non-identical twins, or fraternal twins. An identical twin, in science, we call identical twins monozygotic twins. Now, the reason why we call them that is the way identical twins are two twins that are born at the same time, and they're 100% genetically identical. They are exact genetic replicas of each other. Essentially, they're clones of each other, which we'll come back to here in a minute. But the way that works, because remember, we learned earlier that there are several ways of randomization, several techniques of randomization that cells use to make sure that each one of their children are different, right? You had the random alignment, and you had crossing over, and you had random fertilization. And so the whole goal of that whole system is to make sure that every zygote is different. And in monozygotic twins seem to break that, but they don't. Here's what happens with monozygotic twins. You've got one egg here. Here's my little egg, and it gets fertilized by a single sperm, just like it's supposed to, okay? So you wind up with this fertilized egg called a zygote, which we talked about earlier. And then it starts growing. So the little sperm fertilized it, okay? So now it's going to start growing. So you get a cell and a cell and a cell, right? It's going to start growing into this little bundle of cells here. This is going to be our little embryo here. And then something happens naturally where this little bundle of cells, as it's growing, accidentally breaks into two pieces. We're going to come through here and accidentally break our little embryo here into two separate pieces here. Now, notice these two pieces, all of the cells in these two pieces came from the same fertilized egg. So each cell in both of these chunks is 100% identical to every other cell in both chunks. They're all copies of each other. So if each of these halves go and implant separately in the uterus, they will both develop into babies. But since both of them came from the same sperm and same egg, they're going to be 100% genetically identical. So that's why identical twins are known as monozygotic twins, because they're twins that came from the same zygote. Mono means one. So one zygote twins. One zygote, one fertilized egg, turned into twins because as it was dividing and creating multiple cells, it accidentally broke in half and created two children. Now that's depending on what phase this happens again, right? as long as it happens in an early stage of the pregnancy, the break in half won't hurt it. And so you just wind up with essentially two bundles of these identical cells implanting in the wall, and you create two babies simultaneously that are 100% genetically identical to each other. That's monozygotic twins. Fraternal twins are known as dizygotic twins. And dizygotic twins, if you know the term fraternal twins, they're twins in that they were born from the same mother at the same time, but they are not genetically identical. And the way that one works, di means two, so these come from two separate zygotes. Normally, a woman ovulates a single egg per month, and it typically alternates which ovary is ovulating. But occasionally, a weird fluke happens where both ovaries fire in the same month, and so each ovary releases an egg. So you wind up with two eggs being released instead of the typical one. And if both of those eggs get fertilized by sperm, then you will wind up with two separate zygotes. But since each zygote went through its own randomization processes and each one got fertilized by a different sperm, both of these zygotes will implant in the wall and grow into a baby. But since they both developed with each with its own randomization process, each one's going to be genetically different. So dizygotic twins are different because they came from two different zygotes. It's essentially two different pregnancies happening at the same time. So that's the difference. Monozygotic twins are twins that came from the same egg and sperm, and so they're going to be 100% identical to each other. Dizygotic twins come from different eggs and different sperms, so they're going to be born at the same time, but they aren't going to be genetically identical to each other at all. Now, the reason why this discussion is relevant to artificial embryo twinning is because artificial embryo twinning relies on the concept of monozygotic twins. Essentially, what we're trying to do in artificial embryo twinning is to deliberately create monozygotic twins. Naturally, this happens and you get identical twins, right? But the whole goal of artificial embryo twinning is to do this on purpose. So what they do is they take the egg and fertilize it with a sperm. This is like in vitro fertilization, right? Which we learned about clear back in week 9. So here's our little egg, and it got fertilized by a sperm, so we've got a zygote, and then we put it in a petri dish, and we allow it to divide and divide and divide and divide and divide, and we come out with a flower. Who knew? You get a bundle of, here's our little bundle of cells, so we let it divide, and then after it's been able to divide several times, we've got this bundle of cells here on the petri dish. Then we deliberately go in with like a scalpel or something and manually come in here and cut the zygote into two separate pieces. Or cut the embryo into two separate pieces here, which is exactly what happens in natural creation of monozygotic twins, except we're just doing it on purpose, like human intervention here. So I've cut it apart so that there are two cells here, or sorry, two bundles of cells here, and then we implant each half separately. So just like it happens naturally with monozygotic twins, artificial embryo twinning will result in identical twins. It'll be two individuals that are 100% identical to each other. So in this way, we can clone an organism. We can create two individuals that are 100% identical to each other, which is whole organism cloning. But keep in mind, and this will be important later, this technique can only be used to clone an individual that hasn't been born yet. Because remember, this relies on there being an embryo to split, right? So you couldn't use, if you were trying to make a clone of me, you couldn't use artificial embryo twinning because I'm not an embryo anymore. You couldn't chop me in half and make a second one of me that's genetically identical. So this only works on individuals that haven't been born yet. And essentially, we're just deliberately making identical twins. That's the whole goal there. So in this case, you're going to create two babies that are genetically identical to each other. And they're going to be the same age. They're going to grow up being the same age and everything like that. That'll also become important here in a sec. So that's the goal behind artificial embryo twinning. Now, why would you want to deliberately create identical twins? Well, maybe you're a very ambitious person and you deliberately want identical twins. I don't know. But for a science application, because this technique doesn't necessarily need to be done on humans, right? We can do it on animals. For science purposes, this can be really helpful in studying something called epigenetics. Epigenetics is a study where we've learned over this past several weeks, and let me write out that term here, epigenetics. We've learned over the past several weeks that your DNA, your genes, determine how you turn out, right? Your genotype determines your phenotype. That's not 100% true. It turns out that we've been discovering recently that there is this whole nother level of regulation called epigenetics. In fact, we learned a little bit about this in week 11. DNA methylation, which is a process that can permanently shut down a gene, is an example of epigenetics, where tags or molecules that we stick to the outside of the DNA can alter which genes are being expressed and in what amounts. There are other chemical tags. There are molecules that the cell can stick to histones that makes them behave differently. There are tags that can be stuck to the DNA that makes it behave differently. So you can permanently turn on or turn off or control which genes are being turned on, etc. And so this external regulation, these external influences that can control which genes are being expressed, is called epigenetics. And the way that we can use artificial embryo twinning to study epigenetics is I can create two individuals that were born from the same mother and have identical DNA. And then we can put them in different situations, right? So let's say we're doing this with mice, and I want to study the effect of eating a certain kind of food on their weight gain, right? So if I were to use two completely genetically different mice on my study and fed one my new food and one just ordinary food, I wouldn't be able to 100% determine if my new food made the new mouse fat, because maybe the mouse that I fed my food to had bad genes that made it naturally fat, whereas my control mouse that I fed the ordinary food didn't. So I wouldn't be able to completely rule out that my food was what was making them fat or not. But if I've got identical twins that are 100% genetically identical, then I know that if I put them in these two different scenarios and feed one the new food and one the old food, then any difference in how they turn out isn't going to be because of their genes, because they're identical twins. It's as if you put the same individual through two different treatments. So that helps us do control experiments and allows us to see, and allows us to remove genetics as a factor in studies for various scientific applications. Another application for this, which would be similar, is like drug studies, where I want to see if a drug is effective. And a lot of times they have to try and find identical twins for drug studies. on individuals because it's really helpful because I can put them into two separate groups and give one twin the drug and the other twin a placebo, which is basically just a sugar pill that they think is the drug, right? And the whole goal is to compare these two individuals and see what the outcome is so that we can rule out genetic difference as being a factor in their outcome and isolate any difference as being from our drug. So those would be benefits to artificial embryo twinning from a scientific perspective. Again, there could just be some people that are really brave and want identical twins. I don't know, but those are applications for artificial embryo twinning. Now, let's talk about the other type. Of whole organism cloning here. And that one's called somatic cell nuclear transfer. Kind of a mouthful here. Oh, and before I move on here, back with the artificial embryo twinning, you'll in your reading, there's a list of like cloning milestones or whatever, and I believe, if I remember correctly, the first one was a sea urchin where they split a sea urchin embryo and it created two different ones, and that was the first time that an animal had been cloned or whatever. That was artificial embryo twinning. So they did artificial embryo twinning starting like 100 years ago. Like they started doing this a really long time ago because it's a lot easier to do than this technique that we're going to talk about here. So this somatic cell nuclear transfer is a lot more of a recent development, maybe in the last like 40 years or so. But artificial embryo twinning, they've been doing it for a lot longer because it's a lot easier to just grow up a bundle of cells and chop it in half and then implant both. It's a lot easier to do than what we're doing here. Somatic cell nuclear transfer is different. So once again, we're trying to create two individuals that are 100% genetically identical to each other. However, we're going to go about this one differently. Remember, with the artificial embryo twinning, we were trying to make two copies of a person that hadn't been born yet. Well, how do we clone me? Let's say we're trying to do Star Wars Attack of the Clones, right? Star Wars Attack of the Clones, they take a person, Jango Fett, right? And they make a bajillion copies of him to make all these super soldiers, right? Artificial embryo twinning wouldn't work because Jango Fett's an adult. So he's not an embryo. You can't split him to make more soldiers. You have to find a way to clone an adult human being. So if we were going to try and clone an adult of me, we need to use somatic cell nuclear transfer. So here's the idea. We need to be able to create an embryo that's 100% genetically identical to me and can develop into a baby, right? Well, here's our predicament. I have none of the cells in my body have the ability to develop into an entire human baby, right? But if we were to take an egg cell from some woman and fertilize it, it's going to be completely randomized and it's not going to be genetically identical to me. So somehow we have to create an egg cell that is 100% genetically identical to me. And that's what we're going to do here in somatic cell nuclear transfer. So let's say this little circle here represents one of my skin cells, right? So here's a skin cell, my skin cell, okay? Because this skin cell, it's going to have a nucleus in it. I'm going to put a little sphere in here. And if you remember, the nucleus of a cell holds all of its DNA. So this nucleus here has all of my DNA in it. Now, but this skin cell has my DNA, but it doesn't have the ability to turn into a baby. Now, over here, we've got an egg cell from some random lady. It was donated by some random lady. Random lady, right? That was very generous of her to donate this egg cell. Now, it doesn't necessarily have to be from a random lady, right? Like we could do this for, like if a parent wanted to have a clone of somebody, then then the wife could donate an egg cell and have this done there. So it doesn't have to be from a random unknown person. It can be from the mother that's going to be the surrogate mother. It can be from the lady that they're going to implant it into. But we got an egg cell from a random lady, and it too has a nucleus right here. So that little sphere represents our nucleus here. Now, this egg cell over here has the ability to become a baby, but it doesn't have my DNA. It's not 100% genetically identical to me. So we've got a cell that's 100% genetically identical to me, but can't be a baby. And we have a cell that can be a baby, but isn't genetically identical to me. And we need to make a cell that can be a baby and is 100% genetically identical to me. So here's how this process works. We come into the egg cell here. Oops. And we come in with a very, very small needle. We put this egg cell underneath the microscope and we hold it still. And then we come in with a very, very small needle and poke it into the cell and vacuum out the nucleus of the egg cell. And we suck it right out. And if you can find a video of this, it's really interesting to watch them do it. But you suck out the nucleus. So you've got what they call an enucleated cell. If you see that term, you probably don't need to know it, but just in case you see it in your reading, I can spell nucleated cell. That just means it's a cell that had its nucleus removed. Okay, so it's a cell without a nucleus. So we've got this egg cell over here that had the nucleus removed. Now we're going to come in and do the same thing with my skin cell. They're going to poke that little needle in there, and they're going to suck out my nucleus. And then they're going to come back over to this egg cell and use that needle and inject my nucleus into the egg cell of the random lady. Now, look what we've got. We've got an egg cell that's capable of turning into a baby that has all of my DNA. It's 100% genetically identical to me. So now we can implant this new egg cell into a surrogate mother and allow it to grow, and it will turn into a baby that's 100% genetically identical to me, which is really cool. Now, note that in this case, the clone won't be the same age as the person that they cloned. Remember, in artificial embryo twinning, we created two individuals that were the same age. They were both babies, and then they grew up and they were the same age. In this case, I'm already an adult, but this cloning process isn't going to create an individual that's my same age and looks the same as I do. It's going to create a baby that's going to grow up and theoretically look like I do when they're my age. So that's a difference between somatic cell nuclear transfer and artificial embryo twinning. Artificial embryo twinning creates two individuals that are the same age and are both babies, whereas somatic cell nuclear transfer can clone an adult and create a baby that's genetically identical to the adult. This is the technique that they did on Dolly the sheep. Now, Dolly the sheep was such a big deal because she was the first mammal, not the first animal, the first mammal that they cloned using somatic cell nuclear transfer. So they took a skin cell from an adult sheep and took out the nucleus and put it into an egg cell from a different sheep and put it into and implanted it into a surrogate mother, and Dolly the sheep was born. Now, the downside to this somatic cell nuclear transfer technique is it's very inefficient. Dolly the sheep, and I went and looked this up earlier today, Dolly the sheep was the only one of 277 embryos that they tried to implant in the particular research experiment that created Dolly the sheep. So they tried to do this whole skin cell nucleus transfer thing 277 times, and Dolly the sheep was the only one that worked. Now, nowadays, they've made a lot of improvements since 1997 when they cloned Dolly the sheep. So now something that I read earlier today claimed that they've gotten up to 70 to 80% success rates. So they've gotten a lot better. This one was in pigs. That 70 to 80% was in pigs, but they've gotten an awful lot better at making this more efficient and making sure that the embryos actually implant successfully and survive. But it's still somewhat inefficient and imperfect. But again, as time passes, it will get a lot better. So advantages for somatic cell nuclear transfer. From a human perspective, somatic cell nuclear transfer isn't as useful as artificial embryo twinning, which isn't in itself all that useful from a human perspective, unless you're deliberately creating research subjects, which is kind of unethical. But in animal studies, this can be particularly useful for several reasons. One, for example, if I'm a racehorse person, I own racehorses, and I have this really prized racehorse that's just like, he's a world champion. He's got all the right traits. He's fast. He's got the endurance. He's got everything a racehorse jockey could possibly want to win races. And yet my racehorse is getting old, and he's not going to be in good enough shape to race anymore. Now I can breed him with other horses to try and create an offspring that has many of his traits, but since it's going to be randomized and he's only going to be contributing half the DNA to the new child may not be as good as the father was, right? So the new kid isn't going to be 100% genetically identical to my award-winning racehorse. And so but somatic cell nuclear transfer, if I did this, I could create a new baby horse that is 100% genetically identical to my aging racehorse. And so when that little horse grows up to be a full-size horse, it should have all the same traits as the adult horse did. And so I would essentially have just given rebirth to my award-winning racehorse, which will allow me to stay in the game. You could also use this in cattle, where if I've got a really good steer that has genetic qualities that I like, and it's got really good breeding stock, I can, and once again, it's getting old, I can clone that steer so that I can create a new baby cow that has the exact same traits as my male that's not contributing traits, or the female that is getting old. I don't want to lose them, so I can clone them so that I can keep that breeding stock going. I'm sure there are other uses than just in breeding, but those are the primary ones that are coming to mind for somatic cell nuclear transfer. And we will, here in a sec, we'll talk about another use for this technique that is a lot more useful for clinical applications and for actually using it in medicine. But that's the technique of somatic cell nuclear transfer. So you've got an egg cell that you took from somebody, and you got a skin cell, which, oh, there's a vocabulary word here, somatic. So we talked, or here in the name, we've got this term somatic. Now, what does that mean? A somatic cell, I guess let's circle somatic cell here. A somatic cell is any cell that is not a sperm or an egg cell. So it's any cell that cannot grow into a baby. It's not genetically or it's not reproductively viable. So an example of a somatic cell would be a skin cell, or a liver cell, or a lung cell, or a nerve cell, or a heart cell, or muscle cell. Any cell that isn't a sperm or an egg, or a zygote, I suppose, is a somatic cell. So the whole point of somatic cell nuclear transfer is we're taking a nucleus out of a somatic cell, meaning a cell that isn't a sperm or an egg, and putting it into an egg. So that's that term there. A somatic cell is any cell other than a sperm or an egg. So that's a somatic cell. Okay. I'm glad I remembered to cover that before I moved on here. Okay, so let's wipe the board here. And we're going to talk about another use here for somatic cell nuclear transfer that's kind of a subset of whole organism cloning. It's not whole organism cloning, but it's kind of like a subset, an alternate application of the techniques that have been developed for whole organism cloning. Let's say let's say I'm sick and I'm getting kidney failure, right? I'm on dialysis, my kidneys are failing, I need a transplant. Now, transplants are very difficult. The whole art of organ transplants is extremely difficult because your immune system is designed to recognize things that aren't genetically identical to you. So your immune system will identify organs that came from somebody else as enemy invaders and attack them, and it'll try and destroy the new organ that you had implanted into you. That's bad. They have people that get organ transplants typically have to be put on immunosuppressant drugs to prevent their immune system from destroying this new organ that saved their life. So plus, it's difficult to get organ donors. A, people don't usually like giving up their organs, and B, because of this whole immune system thing, they have to go through several tests to try and find an organ that's as close to being genetically identical to you as possible, so as to try and minimize the amount of immune response we're going to trigger. And that's really difficult to do. And even if you do find one that's really close, like a sibling, it's going to be imperfect, and your immune system will still attack it. Maybe not as vigorously as it would somebody that's more distantly related, but it would still attack them. However, what if I could fix that by putting in an organ that is 100% genetically identical to me? This technique was not working. There we go. Okay. This technique here is called therapeutic cloning. Sorry, my computer's going a little slow here. Okay, therapeutic cloning. Now, I'll emphasize that this is not whole organism cloning. We're not messed up people that are going to grow an entire individual so that I can harvest their organs for my benefit, right? We're not, like, that's ethically unsound, right? Unless you think it's not, you can debate about it in the chat. But for our purposes here, it is widely regarded that growing an entire person just so that you can harvest their organs is very unethical. So somehow we're going to have to use techniques to try and create a new liver for me, or a new kidney, that doesn't involve growing an entire individual that's genetically identical to me, that I can then steal organs from. Therapeutic cloning, the idea here is, rather than cloning an entire individual, I'm going to clone part of an individual. So I can clone organs or other body parts. So once again, it's not a whole person, it's part of them. So I'm going to do the same idea. I can do somatic cell nuclear transfer here. So I can take an egg cell and suck out the nucleus and implant in my and inject in a nucleus from one of my cells, and then we're discovering more and more different signals that the body uses to tell a cell what kind of career it's going to be. It sends a signal, a certain molecular signal that says, okay, you're going to turn into a liver cell. Okay, you're going to turn into a nerve cell. And so we're finding out these different chemical signals that are sent by the cell to tell cells to turn into various organs. So if I want a kidney, I can put this newly nucleated egg cell onto a petri dish and then give it the molecule, or give it those signals saying, become a kidney, become a kidney, become a kidney, and it will grow into a kidney. And they can do this in a lab. They're getting much better at it. They've made a lot of progress in the last couple of years. The idea is that this cell can grow into a kidney, and that kidney will be 100% genetically identical to me. Essentially, I just grew myself a new kidney. So that's my kidney that we just grew on that petri dish. So when they implant that kidney into me, theoretically, my immune system won't attack it because it's going to recognize it as my kidney because it's got my DNA. It's going to look exactly the same as my kidney. Now, this won't be all that helpful if the original problem was caused by my genetics, right? Like if I've got whatever disease that was causing my kidneys to fail is a genetic problem, then that's not going to solve the problem. Like if I'm a hemophiliac, the cells that produce factor VIII protein, which when it's mutated causes hemophilia, we learned about that in week 9. Those cells that produce that protein are found in the liver. So if I get a liver transplant using therapeutic cloning, it's not going to cure me of hemophilia because it's genetically identical to me. So the new liver is going to have the same exact problem, right? But if I got, for example, if I got infected with a virus that, like hepatitis virus, that's killing my liver, then theoretically I could remove the liver, and assuming I got all virus material out, right? I completely cleared your system of the virus, and I cleared out your dying liver and replaced it with your genetically identical liver, then you'd be healed, you'd be cured. So again, this won't solve genetic-based diseases, but it will solve but it could solve other problems, like if you were just having kidney failure for natural causes, or heart failure, et cetera, things that you would need an organ transplant for in that case. Another thing you can do is to combine therapeutic cloning with other techniques, such as the CRISPR-Cas9 technique that we learned earlier, where if I were trying to fix someone of hemophilia and their liver was dying for some reason or another, I could kill 2 birds with 1 stone. I could clone them a new liver, but before I did, I would take out the nucleus and put it into the egg cell, right? And then I could do CRISPR-Cas9 gene editing to fix the broken gene in there, and then grow that one into a liver. And now it would be 100% genetically, well, it'd be 99.9999% genetically identical to me because I fixed that one gene. And so now you've got a fixed liver that's otherwise genetically identical to me. So for all intents and purposes, the cell would still recognize it as mine. So yeah, those are applications for therapeutic cloning. And I mentioned other body parts because it's not just livers and kidneys and hearts here. I've read about a circumstance where they grew an ear. So somebody that lost an ear in an accident or something, they could clone them an ear and surgically stitch it back on or something like that. So there are other applications for this than just organ transplants, but it's a very interesting field. And it's coming, and there's a lot of it or there's a lot of potential for it coming up here in the future. Another less medically relevant application, and this isn't so much a somatic cell nuclear transfer version of therapeutic cloning, but one idea that they've been working on, and it's coming into market now, actually, I saw that they approved it in Singapore a little while back, is lab-grown meat. The idea is, since we've got these techniques for how to grow tissues, how to grow organs and whatnot in the lab, the idea is, what if you got a somatic cell, like a muscle cell, or even do somatic cell nuclear transfer if you wanted to, from a cow or something, and you grew it on a petri dish and told it to become a muscle. And so it would grow into a muscle. And then you could take a cell from that and grow it into more muscle, and then you could turn it into ground beef. And essentially, it, depending on who you talk to, it would essentially be vegetarian beef, because you didn't kill a cow to get it, right? It's completely 100% lab grown, but it's legit beef, which also is nice because you reduce methane output by not having these large herds of cattle. Now, it's still inefficient. They're still working on being able to, as far as I know, they're still working on being able to grow it on an industrial scale where you've got enough meat to make it not super expensive, but it's beef. And they, as I said earlier, I read something recently where Singapore approved lab-grown meat for sale in markets, and it's the exact same thing as beef. It just didn't come from a cow. Interesting, huh? Anyway, those are the thoughts here. If you've got any questions, feel free to reach out to the tutor or your instructors. And hopefully this video was helpful and interesting for you. Thanks for watching.